How is the effect of light on our brain achieved? In 1998, a protein was discovered, melanopsin, which belongs to the group of opsins, is a protein very similar to the better-known rhodopsin, present mainly in cones and rods and known for over 50 years. This discovery created a revolution in the world of visual and neurological sciences. In general, an opsin allows the conversion of a light signal into an electrical signal by modifying its molecular conformation. The electrical signal generated at the output is processed in the area of the visual cortex of our brain. The storage of information, memory, is accompanied by changes in brain waves which represent the activity of the nerve cell blocks that make up the cerebral cortex. These brain waves are observed via electroencephalogram, EEG. Melanopsin would seem to be responsible for sending to the brain information that is not structured two-dimensionally images but concerning the luminous context within which the image is placed, such as the time of day or the quality of ambient light. Such information can also have effects on behavior such as triggering attraction or repulsion for the image content. This information, while coming from the retina, somehow takes a different path from that which forms the image and is not processed by the visual cortex. Melanopsin is found in a small percentage of retinal ganglion neurons, and the signals produced are mainly processed by the suprachiasmatic nucleus, hypothalamus and pineal gland. Therefore, the intrinsic links with the circadian rhythm of light and with the pineal pituitary neuroendocrine system are of great importance in this context. So we can say that the retina performs a subdivision of the luminous information coming from the outside by sending to the brain two streams of data, one structured in the form of an image and the other, concerning the context of the image, unstructured. These flows are processed in different areas of the brain. Does flashing light cause chemical effects? At the basis of neurotransmission, that is, the communication between nerve cells such as the neurons of the retina and those of the brain, is the membrane potential, which is an electrical potential. It produces a chemical signal when at the level of a nerve end it causes the release of a chemical substance, such as acetylcholine, which is released in the synaptic space and transmits the signal to the next cell. The energy carried by the light is able to recharge the membrane potential and therefore to modify the amount of neurotransmitter released in the synaptic space. The recharging action of the membrane potential at the retina level is mediated by the mitochondrion, the very small energy powerhouse that is found inside all living cells. To understand how light recharges the membrane potential, one has to resort to physics and Semi-Osmotic theory developed over 50 years ago. From the Semi-Osmotic theory we learn that at the cell membrane level there are a series of mechanisms that determine the membrane potential that is generated by an asymmetrical distribution of ions inside and outside the membrane. The asymmetry being determined by micromolecular motors that pump the ions against the osmotic gradient. The light that affects a cell can exert a regulatory action on this membrane potential. What I have observed is that this action is dependent on the wavelength of light. Light with a wavelength between 100 and 500 nanometers causes very different effects on cells than that in the range between 600 and 900 nanometers. There is a lot of literature on this subject and it is growing day by day since the importance of phototransduction mediated by melanopsin was discovered. The measurable parameters relevant for understanding the mechanisms involved in intermittent photostimulation are the wavelength and intensity of the light source, which can be deduced at the retina level through the construction characteristics of the LEDs used. Through EEG it is possible to evaluate the different effects caused on the brain by the different wavelengths of light, with the same power incident on the retina. During my studies I used a blue light 1.5 centimeters from the retina, illuminating it with an energy flow equal to 29 micro, while for the red light the flow had a value of 11 micro, equal to about 1/3. What I have observed at the EEG level is that the ratios are reversed. The red light gives a response that is more than double the response with blue light. Finally, as regards the permanent effects of intermittent photic stimulation, I can say that a certain part of the changes in the behavior of the brain caused during the application of the stimulation and observed with EEG are added over time to electrical brain waves, as we have been able to verify over the course of 6 months in a group of responders. At the same time, I observed improvement in cognitive performance, Rivamid test, and in the state of well-being due to the disappearance of depression, Beck test.